Okay, so in advanced bioinformatics, now we are going to study about file formats. That is also an important thing in bioinformatics because saving all the data that is retrieved and analyzed in a proper standardized structure is important to store, transmit, and analyze it later at a global level. So file formats in bioinformatics are standardized data structures like DNA sequences, protein structures, mutation data. These are designed to store, transmit, and analyze complex biological data. And these formats are significant, important because their significance lies in enabling interoperability between software tools. So when you are using, like we have already studied that analysis data analysis requires involves various software tools. So if you are saving your file, if you are saving your DNA sequence, protein structure, or mutations in a similar format, in a similar file format, it would be easy for you to um, operate it in various softwares, the same file. So you are going to actually operate the same file in under various software tools. So also it enhances storage of data, compresses massive data sets, especially those binary file formats. They greatly compress the massive data sets and enhances its readability from computer and enhances the computer operation speed as well, ensuring seamless data sharing across the global research community. So major three advantages of file formats are pipeline integration, where programs in computational pipelines like SAM tools. SAM tools is basically a utility set to analyze sequence data. They chain operations together. Various operations are linked together, processing input and outputs sequentially. Second is storage optimization, like binary formats. Binary formats कौन से होते हैं? Those which are in 0 and 1 form, which are readable, easily readable through computer programs. They like CRAM. They compress data efficiently, and bigger files are compressed and stored in very less storage space using reference-based compression, vastly decreasing server storage requirement. And this decreased server storage requirement also enhances the speed of the processing of your file. The third important advantage of file formats is global collaboration. So when you are saving your data in one single file format, you are actually standardizing it. So this standardization allows researchers worldwide to cross-reference their variant column format, variant data, or compare faster sequence, like whatever the file is. So faster sequence comparison or VCF variant data comparison, cross-referencing is feasible in this. Various file formats that are actually used in bioinformatics to store, share, and analyze biological data include first of all FASTA, that is very common file format visible on NCBI. All of the nucleic acids and protein sequences can be um, downloaded in this format, and this format is very much standardized for BLAST and other data analysis tools. So this structures with a header line like this one, starting with this, followed by an identifier and optional description. Subsequent lines contain the sequence itself. So features include simple, widely used for sequence alignment, blast searches, and databases. And these cannot store detailed annotations or structural information. So by looking at this, this is one of the side effects or drawbacks of this file format, that you are actually not able to analyze or get any structural information or any annotation detail using this um, sequence file format. The other file format is GenBank, which is said like expressed as GB, and the purpose is to store nucleotide sequences with rich annotation. So that is actually covering up faster formats drawback. Structure includes sequence data, gene features, coding regions, promoters, exons, introns, and references from where this all information is retrieved and divided into various accession, features, origin, definition sections. Major features include that these can store comprehensive information about a gene, and this is useful for genome browsers, gene prediction, and annotations where you are actually predicting a gene's function and studying the annotations. So this is important in and widely used in this sense. Next is protein data bank, which stores 3D structure of proteins, nucleic acids, and complexes, and it contains atomic coordinates, secondary structures, and experimental details as well. So in the previous you didn't know anything about how the data was retrieved and obtained, and protein data bank also shows you experimental details and used in structural bioinformatics, molecular modeling, docking, molecular docking, and visualization. Next, we have GFF, general feature format, and gene transfer format, storing genome annotations in a tabular format. So in this format, we have a table which includes sequence name, its source or prediction method of or database, the feature type like if it is a gene or exon or CDS, and start position, the end position, score, how much similar it is, how much aligned it is, strand, phase for CDS, and attributes like gene ID, transcript ID, all these are stored in this table form. Features include standard format for genome browser and gene prediction pipelines. GTF is a strict variant of GHF that is also used in RNA sequence analysis. Next, we have some additional formats like browser extendable data BED, which defines genomic regions like chromosomes, where the gene is located on chromosome, where it ends and where it starts. It is lightweight, used in genome browsers. VCF variant calling format stores all the variations that can be occurring inside your genome or gene of interest or gene under study, including short nucleotide, single nucleotide polymorphisms, insertions, deletions, and widely used in population genomics and variant analysis if you are studying a wide and larger population size. SAM and BAM alignment maps include sequence read alignments, and they are text and SAM alignment is text based and BAM alignment is binary compressed. Both of these are actually computer readable, and these are compressed, and larger files are compressed in smaller spaces, and so these are storage friendly. FastQ is sequencing reads with quality scores, Q for quality, and these are used in NGS pipelines. Newick and Nexus is that like you have already, I suppose कि आपने phylogenetic trees देखे हैं. So those lines that are visible and drawn as a phylogenetic tree, which represent tree structure, branch length, and their connection to the ancestor, that format is known as Newick or Nexus. This format is available online wherever you are studying phylogenetics. So overall NGS workflow includes reading raw sequence alignments, then SAM files which are converted to BAM files to compress the storage, then sorting of this BAM and indexing BAM, and then computer programs, software programs use this indexed BAM for variant calling, visualization, and downstream analysis. Thank you.